Good evening to faculty members, students, family and friends who are joining us today. For those who may not know me, I am Professor Naidu, Dean of the Faculty of Veterinary Science, and I will be your host for the evening. It gives me great pleasure to welcome you to the inaugural address of Professor Henriette van Heerden from our Department of Veterinary Tropical Diseases, following her promotion to full professorship. I'd also like to extend a warm welcome to Professor Themba Musia, who will be officiating tonight as our Vice Principal. Today, we specifically gather online to hear Henriette's address on how she plans on developing her field of study, being the study of infectious diseases with focus on anthrax and brucellosis, being two extremely devastating diseases to both food production and human safety, and both featuring as controlled diseases in terms of animal management in South Africa. To start tonight's proceedings, I would like to invite Professor Musia to do the formal introduction of Professor van Heerden.
Thank you, Professor Naidu, and good evening, ladies and gentlemen. I welcome you all for joining us in this special occasion. And on behalf of the Senate of the University of Pretoria, I would like to take this opportunity to extend a very special word of welcome to Professor van Heerden herself, her family and friends, executive present here, the Dean of the Faculty of Veterinary Sciences, Professor Naidu, and his deputy deans, Professor Marinda Westhuizen and Professor Dave Mahom, and of course other members of senior management who have joined us this evening. I would also like to welcome colleagues in the Department of Veterinary Tropical Disease and in the faculty, as well as our colleagues who have come to enjoy with us this from far afield. And also our bright and wonderful students. We welcome you here because you always make us proud. Ladies and gentlemen, this is one of those auspicious occasions for any academic. And it's a very distinctive tradition for us in academia. And as I was preparing this lecture, I came across some very valuable information for me that Prof Henriette van Heerden was born in Bloemfontein, and where she completed her secondary schooling as well as her university training in microbiology, biochemistry, and chemistry. And with her postgraduate supervision under our very own and one of the best in the world, Professor Mike Wingfield, she completed a master's in microbiology. She moved to the University of Pretoria as part of the Forestry and Biotechnology Institute, better known as FABI, Postgraduate Research Group, where she completed a PhD in 2001. And during her postgraduate studies at FABI, she gained very valuable experience in microbiology, plant pathology, molecular biology, and fungal taxonomy under the supervision of Professor Wingfield. She also joined ARC, Onderstepoort Veterinary Institute, where she expanded her experience in veterinary science and molecular biology. And she also joined the University of Johannesburg and left and joined us in 2003 and joined the Department of Veterinary Tropical Disease. And she was, of course, under the leadership of one of our own as well, Professor Koos Koetser. And as a senior lecturer in microbiology, she established anthrax and brucellosis research groups in the department. Due to biosafety restrictions of these zoonotic and controlled diseases, the initial emphasis of her research was on molecular characterization. And later, with an appropriate biosafety laboratory at her disposal, the research could focus on improving the understanding, detection, and control of anthrax and brucellosis in wildlife and livestock. And it is actually quite amazing to see how the wellness of large creatures can be improved through these microscopic lenses. And the research probes that involved in are multidisciplinary and include bacteriology, immunology, ecology, epidemiology, and molecular biological characterization of brucellosis and anthrax. And this research has made it possible, to create links, at national and international levels. More importantly as well is the community engagement angle of this research. And she reaches out, by all means, through posters, through brochures, through interactions, so that our people and other stakeholders can be able to learn more and more about these zoonotic diseases, which have a very close relationship, between, animals and ourselves. And this is what we see, as one of the fulfillment of the vision of the University of Pretoria, which is a leading research intensive university in Africa and recognized internationally for its quality, relevance, and impact, and also for developing people, creating knowledge and making a difference locally and globally. As an academic in the faculty, she's involved in undergraduate teaching at basic microbiology, bacteriology, and mycology. And she also has a very deep passion for postgraduate supervision. And for part of the soft skills that she develops, it's in leadership, communication, project management, conflict management, and of course the wellness of people in general. And postgraduate supervision, as I say, is close to her heart. And this is evident from the Invitrogen Science Hero Award in the category Platinum Principal Supervisor that was awarded to her in 2016. But she also supervised 15 PhDs and 25 MSc candidates to graduation. Prof Van Heerden heads up the Anthrax and Brucellosis research groups, which comprises postdoc fellows, PhD students, master students, and of course she published 62 articles in scientific journals and contributions, of 2 book chapters. Inaugural address titled Untangling Multi-Host Transmission and Control Dynamics of Anthrax and Brucellosis. Prof Van Heerden will present to us some of the research conducted on control diseases. She will focus especially on anthrax and brucellosis in domesticated and wild animals, especially in Africa. She will highlight the multidisciplinary approach used to detect and characterize these multi-host pathogens with their transmission dynamics in order to enable more effective control and providing solutions. Despite continuous research of these pathogens, the challenge of untangling and breaking the chain of transmission remains. This underscores the interdisciplinary and collaborative approach that is required to ensure animal and human health. The famed Brazilian writer and philosopher who is known to many, many of us, Paulo Coelho, writes in his book, The Alchemist, when we strive to become better than we are, everything around us becomes better too. And Professor Henriette van Heerden says that her aim is to use the scientific experience and God-given talent in a holistic approach to encourage and inspire people. In so doing, she is not only contributing to making us better as the University of Pretoria, but everything around her universe is improving. And I'm sure that at home, or should I say, when she's with her family, together with her scientist husband, Prof Van Heerden, their two teenage children, Andre and Lara, and even their Dachshund, Bessie. I guess that's their companion animal at home. His names are always so difficult for me to understand. Ladies and gentlemen, I present to you our esteemed colleague and a woman of substance, Professor Henriette van Heerden, and look forward to her address. Ad destinatum persequor. Thank you.
Thank you, Professors Naidoo and Musia, for the introduction. Welcome, colleagues, friends, and family to the lecture on Untangling multi-host, transmission and control dynamics of anthrax and brucellosis. The content of this lecture will address the aims of the research, which are to improve the understanding, detection, and control of anthrax and brucellosis in livestock and wildlife. This research was conducted at the Faculty of Veterinary Science in the Department of Veterinary Tropical Diseases at the University of Pretoria. In this presentation, I will provide background of anthrax and brucellosis, followed by some of the research addressing the multi-transmission, host, and control dynamics of these diseases. As I will be introducing two diseases, I would like to highlight their similarities and differences. So, livestock and wildlife hosts are affected by both anthrax and brucellosis. Both are controlled diseases in South Africa due to their zoonotic nature. Thus, they cause disease in animals and in humans. And vaccines are available to protect livestock against the diseases. They differ as anthrax is caused by gram-positive bacterium with endospores, allowing long survival in the environment, while brucellosis is caused by a gram-negative bacterium. Anthrax is non-contagious and cannot spread from one animal to the next, while brucellosis is contagious and is a herd disease. Anthrax causes sudden death, with the animal looking healthy one day just to find it dead the next, while brucellosis causes reproductive problems, which is only observed when abortions occurs. The aborting mother is still alive and other animals are infected through direct or indirect contact. The primary diagnosis of anthrax is the detection of the antigen using microscopy and culture, while the main diagnosis of brucellosis is serology, which is the detection of antibodies to the Brucella antigen. These two diseases are both zoonotic and affect livestock, wildlife, and humans. Thus, it's important to focus on the mode of transmission to the hosts, since understanding the factors will enable control. So let's start with anthrax. The disease is caused by Bacillus anthracis, a soil-borne, gram-positive, rod-shaped bacterium. The rods have distinctive bamboo or box-shaped rods when viewed under a microscope. The endospore form, which can be seen in the image forming in the center of the vegetative rods, allows the bacterium to survive in the environment for long periods. Anthrax is a disease of animals, primarily herbivores, which include livestock and wildlife, but all mammals, including humans, can be infected. Humans are a more resistant and incidental host. In the environment, the spores are ingested or inhaled by the animals. Virulent factors produced by the bacterium rapidly kills the susceptible animal, which result in the contamination of the environment from the animal carcasses, where it forms endospores again, and the cycle continues. Bacillus anthracis pathogenicity depends on the virulent factors encoded on plasmid. PRX1 contains the toxin genes, namely protective antigen, or PA, the common denominator that forms part of the edema toxin and lethal toxin. The toxins alter the host cell signaling and paralyze the immune response of the host. The PXO2 plasmid is the gene that synthesizes the capsule, which protects the bacilli from the immune system. This diagram shows the complexity of anthrax as it spreads through various factors to infect other hosts, and it is non-contagious. Thus, transmission plays an important role in this disease. This diagram, developed by Dr. Wendy Turner, highlights transmission and shows that it is influenced by the environment, pathogen, and hosts. Through our research, we try to untangle the pathogen diversity, survival, environmental factors, and transmission pathways through vectors. In South Africa, the Inzoatic region includes, from left to right, the Gaap region in the Northern Cape Province and Kruger National Park. As mentioned, this bacterium is a soil-borne and occurs in calcium-rich and alkaline soils, which is characteristic of these areas. Research conducted on anthrax has been in collaboration with Skukuza State Veterinary Services. In Kruger, the endemic region includes the most northern part of Kruger, the Pafuri region, which is between the Limpopo and Luvuvu River. Most anthrax mortalities occur along the river, like Harpy Pan. We investigated the suitable region within Kruger that harbors the Bacillus anthracis spores using anthrax-positive carcasses and environmental variables, which identified the Pafuri region and other regions in Kruger National Park. Soil type, normalized difference vegetative index, and precipitation were important environmental factors ensuring survival of this pathogen. Thus, we know that the northern region areas, as well as the soil and environmental factors towards central Kruger, will harbor the spores. Large outbreaks occur in 10-year cycles in Kruger National Park, as seen on this image in the 1990s, 1999, and 2010. Outbreaks also occurred during the same period in the Northern Cape Province, indicated by the green arrows. We investigated the genetic diversity of the pathogen. Bacillus anthracis has a worldwide distribution and, based on molecular characterization, are divided into 3 clades, A, B, and C. The A clade occurred worldwide, while the B and C clades have distinct geographical distributions. In Kruger, the B clade strains were dominant in the Pafuri region, with the A clade strains occurring in the central part of Kruger National Park during the large outbreak as reported by Smith et al. In 2001. However, since the 1990s, the B clade strains seem to have disappeared and only A clade strains are isolated in the northern and central parts of Kruger. This image shows the diverse genotypes of Bacillus anthracis strains in South Africa. The Kruger strains is in blue, grouping from left to right it groups in the B clade, as well as the stern, or otherwise called ABR2 subclade of the A clade, and the dominant ancient A, or ABR56 subclade. Bacillus anthracis strains from Northern Cape Province are indicated in yellow, and those in white is from the Valbos National Park, which was decommissioned in 1997 in the Northern Cape Province. The Northern Cape Province strains group in various subclades in the A clade and are very diverse. In the red box is strains belonging to the same genotype from Kruger National Park, Northern Cape Province, and Valbos National Park. We investigated possibilities on how these genotypes occurred in these regions separated by distance. We did this by investigating the dissemination to susceptible hosts and confirmed that blowflies in Kruger National Park play a role, as the flies feed on infected carcasses and regurgitate their meals on leaves and vegetation that are ingested by susceptible hosts such as kudu and impala. Kudu obtain it through browsing and impala mainly through grazing. These results indicate that blowflies can disseminate Bacillus anthracis within a region, but not over large distances. Thus, we investigated the role vulture might play in the dissemination. Our findings need future investigation, but preliminary results indicate that vultures can travel 250 kilometers and thus have the potential to disseminate spores over large distances. But without scientific evidence of the spread of the spores, it is all thus speculation at this stage. We isolated high diversity of genotypes from vultures not previously isolated in Kruger National Park. In an outbreak, we reported that vultures dilute the spores in the environment when they feed on the infected carcass with a small biomass. As seen in this footage of an infected impala during an outbreak, it was consumed by vultures within 20 minutes, thus reducing or diluting the spores in the environment. In contrast, an infected elephant carcass results in the spread of the disease during an outbreak due to the accumulation of large amounts of vultures. Thus, vultures and blowflies can play a role in disseminating Bacillus anthracis. While the genotypic diversity can be determined from susceptible hosts, most resistant hosts like a hyena, lion, and wildlife, we use the detection of anti-anthrax antibodies in the carnivores to identify the presence and absence of Bacillus anthracis. This was done in Zimbabwe in collaboration with University of Zimbabwe in Surat. The serological methods were established in the laboratory for a vaccine development project and were used towards more than one aim. Thus, we know transmission in South Africa is influenced by high genetic diversity in the pathogen, with higher mortalities in kudu and impala in different seasons in Kruger National Park. Vulture and blowflies are vectors that can play a role in the dissemination of the pathogen. And environmental factors such as soil and vegetation index and precipitation contributes towards the survival of the pathogen. Understanding the pathogen and its transmission in the natural system can be used towards management practices. In livestock, a live spore vaccine is available. This vaccine for livestock was developed at Onderstepoort by Max Stern and consisted of a strain lacking the PXO2 capsule encoding plasmid. The Stern vaccine is thus a avirulent live spore vaccine, and in collaboration with Dr. Wolfgang Beyer, we investigated the development of non-living vaccines to replace the Stern live spore vaccine. We focus on non-living vaccines mainly to reduce the use of live spores, as well as non-living vaccines can be used simultaneous with antibiotics, which is not possible with the live spore vaccine. Before discussing the non-living vaccine candidates, we investigated the Stern live spore vaccine with a virulent Bacillus anthracis challenge, where goats were vaccinated once and twice, then challenged, and the protection was evaluated. Goats vaccinated twice with the Stern live spore vaccine provided 100% protection against the virulent challenge, while goats vaccinated once provided 60 to 80%. The difference between the 60 and 80% protection was the time period of the vaccination, thus indicating it takes time for protective immunity to develop. The non-living vaccine candidates were selected based on anti-spore and anti-toxin strategy, consisting of the protective antigen, which, if you can remember, is the common component in the lethal and edema toxins, as well as BCLA component of the exosporium and formalin inactivated spores. This was Stern spores inactivated with formalin. Goats were vaccinated with non-living vaccine candidates 3 times and then challenged. We also evaluated a passive mouse model using AJ inbred mice that are susceptible to the Stern strain. With the passive mouse model, sera from the vaccinated target animals are injected in the mice, which are then challenged with a lethal Stern dose and observed for 14 days for survival. With the direct target animal challenge, the goat group vaccinated with PA, BCLA, and FIS had a higher percentage protection compared to those with only PA and BCLA vaccine formulation. Similar results were obtained with the passive mouse protection experiment than the virulent challenge experiment. The passive model provided 87% protection to goats vaccinated twice with the Stern vaccine, and also provided protection to animals from the non-living vaccine candidates, namely BA, BCLA, and FIS at 74%. Thus, the non-living vaccine candidates provided protection, and we could use the mouse passive protection model in future to evaluate anthrax vaccines. Various other articles have since been published using the passive mouse model to evaluate anthrax vaccines. Immunological data of the non-living vaccine experiment indicated that the anthrax titer of the non-living vaccine after 2 vaccinations, which is indicated by the blue line, was similar to the Stern live spore vaccine titers. Thus, in the follow experiment, we used 2 vaccination protocol with the non-living vaccine candidates without the BCLA. We focused on bovine as feedlots use simultaneous vaccination and antibiotic treatment. The veterinary vaccine market also requires low-cost vaccines, and thus to reduce the cost of the non-living vaccine, a crude PA as well as a purified PA with the FIS vaccine formulation were tested and compared with the Stern vaccine using immunological test and the passive protection model. The non-living vaccine candidates were done in combination with penicillin gene treatment, and protection measures using passive mouse model were used. The passive protection showed that the purified PA and FIS vaccine formulation in the blue line provided similar protection to the Stern vaccine in the brown line. The crude PA and FIS vaccine formulation indicated by the red line provided low protection, and thus the crude PA vaccine formulation cannot be used. Purified PA and FIS vaccine in combination with penicillin G treatment in blue provided similar protection to the Stern vaccine group in green. The Stern group with the penicillin G provided low protection seen with the brown line. Despite the protection and advantages of non-living vaccines, the problem is that non-living vaccines is 10 times more expensive than the Stern vaccine. We do not foresee the commercialization of the vaccine due to the emphasis on low-cost veterinary vaccines, especially as the Stern vaccine provides good protection. I hope you enjoyed the unraveling of the complexity of the multiple host, transmission, and control aspects of anthrax thus far. This brings me to brucellosis. Humans get infected through contact with infected animals and animal products. The main clinical sign in animals is abortion, with the mother remaining infectious. The aborted fetus contains a high number of bacteria and are highly infectious. This disease spread within a herd through direct and indirect contact. It is caused by a gram-negative bacteria of the genus Brucella, which is slow-growing, making isolation difficult. Different Brucella species affect multiple hosts. And this table illustrates the host preference. For example, Brucella abortus, with all its various bivalves, mainly cause brucellosis in cattle, while melitensis causes brucellosis in capron like sheep and goats. Brucella abortus, melitensis, and suis are highly pathogenic to humans and occurs in livestock, and we will focus on these species in livestock and wildlife. Brucellosis can be controlled in cattle using the S19 vaccine. Heifers must be vaccinated before 8 months, and older non-pregnant animals can be vaccinated with the RB51 vaccine. Bulls should not be vaccinated as the vaccine could cause infection. Goats and sheep rams are vaccinated with Brucella melitensis Ref1 vaccine. The most common available sample is serum that is used in serological tests. In a screening test, the roast Bengal test or RBT are used, and the RBT positive samples are confirmed with complementation fixation tests, the CFT, or the indirect ELISA. In South African veterinary laboratories, the RBT and CFT tests are used in series. The two serological tests do not ensure 100% that the tested animals is brucellosis free, and thus repeating tests should be used before a free status can be obtained. Culture is the gold standard, but lacks sensitivity. Uterine discharges and milk samples can be used from live animals for culture, whereas various tissues can be used for culturing from dead animals. Brucella culture must be identified to species and biolevel using a series of biochemistry tests known as biotyping, and it's very time-consuming. Problems also exist with biotyping. For instance, with Brucella abortus biovar 3, most strains require carbon dioxide for growth, but some do not. If you have an abortus strain of this biovar that does not require carbon dioxide, it can easily be confused with biovar 6. Through biotyping, it is not a perfect system. It's currently the only typing method available. As mentioned, farmers or animal owners become aware of the problem when there is an abortion. And abortions are caused by various microorganisms, and the causal agent must be identified. Abortions caused by brucellosis occurs in the 5th and 7th month of pregnancy that coincide with reduction in milk production. This disease causes several reproductive problems such as stillbirth, weak calves, retained placenta, decreased fertility with poor conception rates. Reproductive problems also occur in bulls and rams. And the biggest problem is some calves from infected cows may have latent infections, and these calves will become positive when they become pregnant, abort, or give birth. This information indicates that human brucellosis has been reported since 1871 onwards, with Brucella abortus and Brucella melitensis isolates reported from humans. Animal brucellosis was reported in sheep and other livestock, also with isolation reports of both Brucella abortus and melitensis. The first published outbreak of Brucella abortus occurred in 1906 in Johannesburg. Thus, brucellosis has a long history in South Africa. Brucellosis is a controlled disease in South Africa, and the bovine brucellosis scheme was gazetted in 1988, enforcing the vaccination of all heifers with S19 vaccine, testing of high-risk animals such as dairy and export animals, with the main emphasis on bovines and slaughtering of positive animals. The scheme has limitations as it is voluntary and mainly focused on bovine, but has been revised in 2017 by the department with recommendations. Although brucellosis is a controlled disease, limited information is available. Initial seroprevalence studies in KwaZulu-Natal at the abattoir and also in communal cattle showed low prevalences of approximately 1.5%. Later, high prevalence of 9.2 using a less sensitive culture method was reported in cattle. In sheep and goat, various outbreaks of Brucella melitensis has been reported since 1965, with the latest in 2015. Brucellosis was first detected in humans in most of these outbreaks, and upon investigation linked to infected sheep and goats. Thus, Brucella melitensis was isolated from sheep and goats as well as from humans. To control any disease, surveillance is necessary of susceptible hosts, as well as to determine the mode of transmission and risk factors. This was done in a project initiated by Prof Henriette with the ARC OVR and Gauteng government at abattoir. We started by determining the seroprevalence of samples submitted from 2007 to 15 at the OVR laboratory and found 5.85% in all livestock and 6.3% in tested cattle. We found similar sero and culture prevalences from slaughter cattle at the Gauteng abattoirs than the respective data from the OVR laboratory. Brucella abortus bivar 1, Melitensis bivar 2 and 3 cultures were isolated from the cattle tissue and biotype. This was the first report of Brucella melitensis in cattle. We confirmed the presence of Brucella melitensis and ovis using PCR assay in culture from sheep tissue collected from the abattoirs. As abattoir workers have direct and indirect contact with these animals and are at risk, we developed the education material on zoonotic diseases and started awareness by educating the abattoir workers on zoonotic diseases and their risks. Education is key, and as educated people, we should make it a priority to educate. With the isolation of Brucella melitensis from cattle, it indicates the spillover to other livestock can occur due to farming practices in the country. This can also occur in wildlife in South Africa. In South Africa, waterbuck, hippos, zebra, eland, sable, impala, and the African buffalo have all tested brucellosis positive. We published an investigation into outbreaks of Brucella melitensis in sable on our game farms in South Africa, in where we investigated the outbreak source and mode of transmission. In 2004, melitensis outbreak occurred in sable on a game farm with other wildlife as well as with sheep and goats. The farm was quarantined and wildlife as well as livestock were tested on the farm. None was positive for brucellosis other than the sable. Before the outbreak, you can see that the sables were bought and sold, such as in 2001, sable bought from Farm 2. All the sable tested positive were euthanized and sable were continuously tested. Despite the preventative measures, outbreak occurred again in 2013 and 14. Before the 2013 outbreak, sable was again purchased from Farm 2. With each outbreak, sable on Farm 2 were tested and all were negative for brucellosis. Notice that before the outbreak, sable sold to other farms like Farm 7, 8, and 12, also all tested positive. In this investigation, we could not determine the source, and despite testing for brucellosis, outbreaks occurred again. Heifers were purchased and tested, but from a farmer that does not routinely test. Thus, latent infection could not be ruled out. On another farm, Farm 4, Brucella melitensis outbreak occurred in sable in 2016. This farmer also purchased sable from Farm 2. This investigation indicates that in these outbreaks, the farmers most probably purchased infected animal, highlighting that control management should include purchasing animals from brucellosis-free farms, which requires regular testing. We reported similar findings when we analyzed a whole genome sequence from Brucella abortus isolates from 2 brucellosis-infected farms in South Africa. The disease can spread within a herd, as seen in Farm 1, as the same genotype was isolated from cattle. While on Farm 2, it seems that brucellosis were introduced by infected animals, as different genotypes occurred on this farm. This brings it to the end of multi-transmission, host and control dynamics of brucellosis. To conclude, anthrax and brucellosis are 2 different diseases, but control of these diseases are complicated by the fact that the diseases infect multiple hosts, which include livestock and wildlife. Although anthrax and the brucellosis research group at the University of Pretoria will further untangle the complexities of these diseases, it is essential to emphasize the use of existing vaccines to protect livestock, which will protect humans. Animal and human health can be ensured by educating people on these diseases and should be a priority for all research. Before I end this lecture, I would like to thank and acknowledge, First of all, God, the creator of the universe, all the glory forever and ever. My parents, Dries and Sinta Brits, that gave me the opportunity to study further. My wonderful husband, Skalk, and our children, Andre and Laura. Thank you for your love and support, and I love you. Thank you to my family and friends for being part of my life. I wish to thank 3 mentors that influenced my scientific career with their vision and passion. Mike Winkfield was my postgraduate supervisor and a man of great vision that established FABI at the University of Pretoria. I was privileged to be part of the first postgraduate students when FABI started. Thank you, Mike, for challenging and influencing me. My first job was as a scientist at the ARC OVR, and Basil Alsop was head of the section that sequenced the Ehrlichia ruminantium genome. This was before next generation sequencing using Sanger sequencing. Basil showed me that with a dream and a team, you can do much. Koos Koetse, that was head of veterinary tropical diseases when I started in 2008. He influenced me to train future leaders on diseases in Africa. Thank you to all the collaborators that contributed to research with me and my group, and I hope that many more findings will follow in future. Thank you to all the colleagues, which include academics, admin, and support staff at the faculty, but especially those at DVTD. I might be presenting the lecture, but the true stars behind this research are the postgraduate students. I applaud you and thank you to each of you, as I'm blessed to be part of your life. Thank you to everybody that helped with the inaugural lecture. And lastly, thank you to all the supporters and funders of the research.
Dear colleagues, we have come to the end of tonight's function. Unfortunately, with the event being online, we are unable to fit in a live Q&A session. However, if you do have any questions from tonight, please feel free to drop an email to Henriette, as I'm sure she'll be more than amenable to respond to them. Finally, before we close, I would once again like to congratulate Professor van Heerden on her promotion to full professorship. As we just heard in her inaugural address, she has much to do in the further management and eradication of 2 economically devastating diseases in the country. With the major strides she's already achieved in the field, as well as her wide networks of collaboration, I have no doubts we'll see many more major achievements in the field. I also look forward to seeing her new anthrax vaccine get market authorization and bring the faculty to the fore of veterinary vaccinology. With that, colleagues, I wish you a very good night and goodbye. Thank you.